Welcome to the Picture Language Seminar. We're very happy today to have Kushal Seetharam. Kushal just finished his doctorate at MIT over the summer, and now he's a postdoctoral fellow at Harvard. He'll tell us today about quantum simulation of NMR experiments. It's a very interesting topic, and Kushal, we're looking forward to your talk. So please share your screen, and the floor is yours.
Perfect. Thank you, Arthur. Let me go ahead and let's see, hide. There we go. Is it clear? You guys can see?
Absolutely. Perfect.
Beautiful. All right. Hi, everyone. I'm going to talk today a little bit about a potentially practical context in which we can use quantum simulation to help in mostly biochemical NMR experiments. And I'll go through and explain what that is and where we may try to get an advantage, not a complexity theoretic one, but perhaps a practical one using quantum devices. So just as an overview, NMR spectroscopy is kind of a very widely used tool in a variety of subjects, including biochemistry, materials chemistry, and a host of different other areas. And mostly today, I'll be focusing on kind of a biochemical context, but many of the ideas, especially when I talk about solid state NMR, apply to materials chemistry as well. So the picture to have in mind of these experiments is, let's say I have some very large superconducting magnet, that can output a field of several Tesla, and I put in a sample in it, let's say some set of molecules either suspended in liquid or solid, and I look at the spin dynamics of the system as they interact with each other and measure the average magnetization of the nuclear spins. And from this time signal, what I'll end up getting, once I Fourier transform it, is some sort of spectrum. And from the spectrum, I want to learn about whatever was in that sample, whether liquid, solid, whatever it was, and infer something about its chemical structure, function, dynamics. And the NMR community is actually quite old and mature and has developed all sorts of sophisticated ways to, even with just such a simple experiment, in principle, to extract a ton of information about the samples they want to learn. So we can actually frame this formally, as some sort of inference problem, where I have, let's say, a molecule and I look at its spin dynamics, and there's noise in the system itself. These experiments happen at room temperature typically. And there's different decoherence mechanisms. And so I look at the noisy spin dynamics of the system, and then from that I want to infer something about the molecule. So in this case, I want to talk about, focus mostly on how simulation of these spin dynamics can be useful. That's not true for all NMR contexts. There's many cases where there's series of sophisticated heuristics where they can look at just the spectrum itself and back out the information they care about. But there are many interesting contexts where simulation can actually be a very useful tool to help in this inference problem, as long as you can compute the spin dynamics. And a subset of those contexts also are cases where the simulation can be hard to do classically, even with the best software that people have developed so far. So this is kind of the crux of the idea of this talk, is that why can quantum devices perhaps be helpful? And it's because quantum devices themselves, these simulators and computers, are noisy spin systems, and perhaps they're very naturally suited to simulate other noisy spin systems. And this is really kind of the hope and intuition underlying it. All right, so to start making things slightly more concrete, the observable that's computed during the NMR experiment is called a free induction decay. And really, we can think about it as an infinite temperature two-point correlation function for people for who that language is familiar. Otherwise, we can think about it as just the average magnetization of the spins in the sample as a function of time. And of course, this is where we pick up effects from the interactions between the nuclear spins. So we first, because we apply such a large magnet, the spins are polarized, let's say in the z direction, which is the direction the magnetic field is applied. And that's where that SZ total comes from, the total spin. Then you undergo time dynamics and then measure the transverse magnetization through a set of RF coils. And this is the signal you get, a time signal you get. And a Fourier transform of this gives you a spectrum. And there's all sorts of complicated protocols you can do in pulse sequences to try to encode more information in the spectrum you get. The Hamiltonian that generates the dynamics, a typical one looks like this. Well, in this case, let's assume I'm speaking about spin halves, which is a large amount of NMR, but later on I'll actually talk about some experiments where the spins are 5 halves, 7 halves, something larger than that. But just so we can understand like how the dynamics tells us about the structure and function of the molecule, we can look at these terms. And for example, this local sigma z term, which in condensed matter we might call a disorder term or something like this, corresponds to the local response, magnetic response around particular nuclei in the system. Let's say I have these hydrogen protons, which is the nuclei that I'm measuring in my NMR experiment. The electrons surrounding them will screen the external magnetic field corresponding to what that local electronic structure looks like. And because the electron energy scale is several orders of magnitude faster than the nuclei just see kind of like an effective screened magnetic field. And this is called in the literature chemical shift. And this shows up in the nuclear Hamiltonian just as a local field. And this Heisenberg exchange, this S.S term, actually arises from a bond-mediated interaction between nuclei, where both the nuclei and the electrons that couple them in the different atoms into bonds need to satisfy Pauli principle essentially, and from this you get some exchange couplings. And if you know these parameters, they tell you about the geometry of the bonds, the bond angles, and you can put constraints that way. The last term I've shown here is a direct dipolar coupling between the nuclei, and of course this depends on the relative positions of the different nuclei, and so hence can tell you about the structure of the system. So this is just an example where the different parameters of the Hamiltonian themselves, if you can learn them from back them out from the spectrum, give you information about the geometry, structure, and then depending on your protocol, the dynamics itself of the system you're studying. And this is what you're trying to learn. Though depending on the zoo of NMR contexts and which one you pick, some of these terms are more relevant than others. Some of them get averaged out. There's complications that arise from orientation dependence, which I'll get into later. But this is the basic idea, is that I can almost view NMR as a Hamiltonian inference problem if I'm simplifying it to an extent. So let's talk now about where simulations can be useful in this inference problem and perhaps difficult. And what I'm putting right now is very much a heuristic picture. Don't take it as anything quantitative. And this is just based on kind of intuition I have by looking at the literature. One NMR context is metabolomics. These are very small molecules, which you can typically represent by, let's say, like on order 20 spins. There are many of them, and they're very important for understanding various biological and biochemical functions. But you don't really need simulation to understand these experiments, NMR on these small molecules. The heuristics people have, and comparing them to lookup tables of other data they have, actually turns out to be quite sufficient. And even if you wanted to do simulation, because the systems are so small, the classical simulation software will do it very well for you. The one interesting thing that I've seen some people floating, though I don't know what the metabolomics community thinks at large, is that a big problem in metabolomics is there's a large number of these compounds that people just don't have time to actually screen and investigate and experiment. So there are suggestions to maybe use simulation to do high throughput screening, like in silico, like just by examining their properties via simulation. But even if you wanted to do this, as I mentioned, you likely don't need quantum hardware to look at this problem. going kind of further up, both usefulness and difficulty, you have biomolecular NMR. And in this case, I'm actually specifying to say, let's say liquid state NMR. Here, simulation, there are sets of heuristics that people use in experiments and sophisticated methods to back out what the Hamiltonian parameters they care about are from the sets of spectra they have. But usually this is like one quite time consuming, requires several experiments. And simulation can still be nice to confirm the parameters they get, right? So let's say I use all my heuristics using various multidimensional NMR experiments. Then if I can take those Hamiltonian parameters, run it through a simulation and see that the spectrum I get matches what I got in my experiment, then I have better confirmation that the numbers I pulled out are correct. Because usually these heuristics are derived from various approximations. So in this case, the difficulty of the experiment varies based on the protocol and context as far as how to simulate it. So for liquid state protocols where you largely only care about coherent dynamics in the system, which are typically, let's say, these exchange couplings, then classical software actually does quite a good job. And the reason is that there's enough kind of decoherence in the system where you don't need to keep track of, let's say, the entire Hilbert space that's there. And you can, truncate, your basis in your time domain simulation, so on and so forth, and actually manage to kind of efficiently, simulate these experiments, using your favorite supercomputer or cluster or whatever it is. The situation changes when dissipative dynamics ends up playing a role in the NMR experiment. These are cases where that dipolar interaction term I showed, which normally in liquid state NMR average out because of rapid rotation of the molecules in the system, ends up coming into second order as a dephasing essentially. And when you want to simulate these experiments, it actually can become quite difficult. And this is also a very practically relevant context. These types of relaxation experiments are one of the most commonly used tools to study large proteins and other biomolecules that are relevant not just for understanding various biochemistry processes, but also for drug development and pharma applications. So these are cases where, again, there are sophisticated heuristics and repeat experiments that can be done to extract out that information without simulation, but simulation would be one, very nice, and to confirm those parameters. And you can actually imagine if you had such a black box to do these simulations, you might, if someone cares, I can talk about this a little bit later, come up with new ways to actually screen compounds and do structure identification by dramatically reducing the amount of experimental effort that's needed to do this. So that's one context. Other is electron spin resonance and electron paramagnetic resonance. These are cases where you have, it's rather than using nuclei and doing magnetic resonance, spectroscopy on them, you do it with electrons instead. This is one context, actually, I'll talk about more in detail later. But these are cases where you actually only have maybe a handful of spins. So you don't have many spins in your system, as opposed to, let's say, in a protein that I mentioned before, you might have thousands. In this case, maybe you have one, two, a few, but the experiments can be challenging because instead of having spin halves, sometimes you might want to use higher spins, 3 halves, 5 halves, 7 halves, and there tends to be kind of an obscene amount of ensemble averaging that needs to happen to model the system. And these are experiments in which, without simulation, you cannot interpret the experiment at all. So simulation ends up being very useful in this case. Another is solid state NMR. So solid state NMR, I will actually talk about more in detail later as well. These are cases where that dipolar interaction I mentioned does not average out to 0 because of rotation, because there's nothing that's rotating. There's still broadening that comes in the system, but you both have to kind of include these long-range dipolar interactions, which makes the dynamics quite difficult itself. But then you also have to do some degree of ensemble averaging over the different, disorder configurations of grains in your sample. And so this is another context in which you actually people find it very hard to interpret their experiments without using simulation. and a simulation can actually be quite difficult where classical simulation software can borderline do it in some contexts, but it becomes practically challenging. So if we're looking for a place where you want to use quantum devices to help accelerate this application, we probably want to kind of look in these contexts and try to identify what are perhaps the common characteristics of these difficult and useful contexts for which we might want to target with our quantum devices. So the first characteristic, as I alluded to, are cases where the dynamics itself is hard. This is the normal application context of why people think quantum simulation or Hamiltonian simulation might be one of the first practical uses of quantum computers and simulators. And the idea is, simply that if I'm trying to simulate the quantum dynamics of a system, perhaps doing that with a quantum device is more natural than doing it with the classical computer. Of course, like we have very sophisticated classical algorithms to do this, so we have to be very careful when claiming or hoping for a practical advantage, especially on near-term machines. The second case, the concrete examples for where this dynamics is hard in a very loose sense, not I'm making complexity theory statements here, but is one, if you do these relaxation experiments on large biomolecules, which this is this nosy nuclear Overhauser effect spectroscopy, that can be difficult because as well as solid state NMR, where that dipolar interaction comes in as a coherent effect as well. So the second is a case where these simulations are both necessary and difficult, or where you have to do an appreciable amount of ensemble averaging. And that's actually going to be the main focus of the ideas I'll talk about later in the talk. But this is also a case which shows up several times, for example, with small spin, systems of a few number of spins, but perhaps higher spins, which is ESR and EPR. And then solid state systems where you both have a large number of spins as well as the necessity to do ensemble averaging. So these are kind of the characteristics we'll try to target with our quantum simulation. So first, before we get into the ensemble averaging itself, let me just explain how you might compute the spectrum itself. So that first thing, that given a coherent an NMR experiment described by some coherent Hamiltonian, how do you actually compute the observable they measure in the experiment, and then use this to get a spectrum. So we can write down, in the simplest way, the NMR spectrum is this A of omega. It's a Fourier transform with some finite line width of this free induction decay S of t. This S of t is a average magnetization as a function of time, which I've written down here to the right. I can now expand that SZ total, which was the initial state essentially of the system in a high temperature or high field approximation, into its eigenstates, which I'm calling M of j. And then just kind of incorporate the time evolution from there, basically from the top line to the bottom, switching from, I guess, Heisenberg picture to Schrodinger picture for people who are familiar. And then what I can see is like really this algorithm or computation of this correlation function S of t really just requires me to prepare computational basis states of my quantum device, which are eigenstates of SZ, and time evolve them according to my desired dynamics, and then measure the average magnetization in a particular basis, which is, of course, again, just a computational basis measurement. I can be turned into that with the appropriate rotation. So this is something that's very natural to do on a quantum device, as long as we have the ability to implement the unitary dynamics, right? You can, if you're astute and paying attention, you can ask me, am I not cheating? Because this summation I've written on the right-hand side actually has an exponential number of terms and the number of spins. But it turns out you don't need to actually compute every term in that exponentially large sum. You can use sampling to reduce the cost. You can even do clever things like importance sampling. And rather than being 2 to the n, this ends up, you end up being able to do it with about n squared samples. So this actually scales polynomially and is not exponentially difficult to do. So just to prove this works, this is not the main result of the talk. It's prior work that we put out, but we did an experimental demonstration on trapped ions of a very small molecule, just 4 spins, without zero field experiment, which I won't get into. But the green line here is an actual zero field NMR experiment on this molecule acetonitrile, or really the methyl group of it, which is this carbon and 3 hydrogens. And the blue line, or bluish blackish line, that's sitting on top, is the computation on the ion machine. And there are kind of a lot of tricks that we had to play to get this to come out with such a nice resolution. But you see that the 2 main peaks, spectral peaks at J and 2J, we kind of capture with almost quantitative accuracy. There's this spurious peak at J over 2, which really turns out to be a result of both decoherence in the ion machine plus some very interesting set of coincidences of the particular system we were studying and its symmetry. And if people are interested, I'm happy to get into it, but it's not a problem if you were to simulate other systems. But this is just to show that you can compute, so that algorithm I mentioned, can compute the spectrum and compare it quite accurately with realistic molecules. All right, so now let's talk a little bit about the second characteristic I mentioned, which is is there a way we can do ensemble averaging for various NMR simulations with a quantum device that might have an advantage in some way over kind of parallel simulations on classical machines? And there's 2 approaches and ideas I'll talk about. One is both are using ancillary degrees of freedom in your system. One using discrete ancilla, let's say ancilla qubits. The other using a continuous degree of freedom, a bosonic ancilla. And I will say that these ideas I'm talking about are very much works in progress right now. So this, take that with as it will. This is kind of I'll sketch out the ideas, but we haven't worked out every detail yet. So happy to talk about any questions you have. So the first context I'll talk about with these discrete ancillas to give us a concrete place to start. Let's talk a little bit about solid state NMR. So solid state NMR is also a widely used tool to study pharma relevant proteins. You can look at membrane proteins, for example, the spike protein in COVID. You can look at polymers, industrial catalysts, battery materials. There's a host of different systems that are studied with this technique. And often you need simulation to really infer the chemical structure or the information you're understanding of the system. And one of the challenges with this is that the main interaction in the system is this long-range dipolar interaction, even at higher fields. And of course, while there is a fair amount of broadening in the system because of the different orientations the different Prouder grains in your system take, this still generates a fair amount of correlations due to the long-range character of the interaction. The second challenge is that in any of these experiments, you don't have one copy of the system. You have 10 to the 12 of them, all sitting with different orientations with respect to your external magnetic field. And what you measure in an NMR experiment is actually an ensemble average over all of these, right? So this is the so-called powder average for people who are familiar. And when you need to do simulation to match these experiments, as I mentioned, one aspect that's challenging is the long-range dynamics itself can generate appreciable amount of correlations. And the second is that I might need to average over 1,000 or 10,000 angles in orientations of the system. And so if I'm doing this with classical computing, these are all independent simulations, of course, but I need to run that separately, and this can quickly get quite onerous to do. And the hope for quantum advantages in this case is that perhaps I can more naturally encode these dynamics on a quantum computer or simulator. And the second is that, as I will explain now, there's ways you can try to use ancilla qubits to do the entire ensemble averaging in essentially a single pass. So with the same cost it would take to do a single computation of a spectrum of the sort that I just showed you. So let me sketch out that idea very quickly. This is kind of the Hamiltonian to keep in mind. Usually you operate in a high field case where you would, this Hamiltonian becomes a little bit simpler, but I'm going to keep it this way for technical reasons. If you're curious, I can explain later. We have some large Zeeman term, which is this omega naught coming from the external field, these local chemical shifts, omega sub I, and then the dipolar interaction, where the dipolar interaction constant is, of course, related to the distance. And these Eij, omega, big omegas, those are the dipole vectors between different spins in the system. And they encode really what the orientation of each molecule is. So this is the idea of what we want to do, but not the concrete implementation. The idea is very much that if I have in the bottom line rho naught, which is a set of n spins in the system, and then I have a set of m ancilla, if I put the ancilla in an equal superposition, and then depending on the state of the ancilla, I can apply different unitary dynamics, different Hamiltonian, each one corresponding to a different orientation. And then I just measure the spins at the end, my standard measurement that I explained before, while ignoring the ancillas, tracing them out. What I will get is exactly an equal average, ensemble average over all the different orientations of the system. Of course, this is nice to say, but kind of meaningless in practice, because what I've written down or shown you here is a many-body unitary controlled by many ancilla qubits. And of course, implementing this in a generic case is extremely difficult. So this is only useful if we can figure out an efficient construction to do this in practice. And that's kind of what I will show you now. So before we get to kind of the full construction, let's for the full Hamiltonian there, let's assume I just had the dipolar interaction. This makes things a little bit simpler and is easier to explain the idea. Then what I really want to do is physically in my NMR experiment, I have 10 to the 12, let's say, grains, powder grains, each one a copy of the system. And the orientations I'm trying to average over have each one shifted with respect to the external field. But of course, this is just a one way to view the orientation. I can either view the molecule is fixed, sorry, the field is fixed and the molecule changing orientation, or I can put myself in the molecule's perspective and view the molecule is fixed and changing the field, right? So this is just an active versus passive transformation. So we're going to exploit that idea here and say, hey, okay, I define the unitary dynamics of the system for some fixed orientation of the molecule omega. Then let me apply a global rotation on the system, controlled on the set of ancilla qubits, that will then, as far as the molecule is concerned, just change the direction of the external field. And if you do this, this turns out to actually be efficient, because these are just global spin rotations, which can be decomposed into just n independent 2-qubit rotations between each ancilla and each of the system qubits. And if you do this for m different orientations, you'll get functionally what I described earlier, right? So now let me kind of put back the full Hamiltonian, right? So now we also have these chemical shift terms and local sigma z's, and the complication is, of course, that these terms of the Hamiltonian will not commute with those spin rotations. So a way to get around this is that we can now change what was just the simple dynamics of dipolar interaction over the system. We can now trotterize it, for example, discretize it in some way, split it up into chunks, and then basically fix the chemical shifts, right? By putting in essentially the same type of controlled rotations to include those terms. And because we've discretized the dynamics, you're suppressing the non-commutation with the number of steps L that you've chopped it down into. But the key thing is, of course, that these V operators, V of K or whatever, are also just spin rotations that are controlled by individual ancilla. So the same way as those R rotations were composed of just n times m 2-qubit gates, that's the same case here. Each of these V's will be the same as well. And so you have essentially a kind of simple construction on how to do this ensemble averaging, and in principle you can therefore do efficient powder average solid state NMR simulations. In practice, of course, this is not likely to be a immediate near-term application, in the sense that you might want to do 100 spins with some large number of ancilla, but it perhaps is one of the earlier applications you'll get to as you develop these devices. And you can even think of potentially analog routes where, for example, Rydberg atoms, if you put them in their S and P states, have natural dipolar interactions. And so then, you can try to avoid some of the cost of implementing this simulation digitally. And then also use similarly, some blockaded interactions between the Rydbergs and the ancilla Rydbergs and the main system to do this superposition trick. So it is somewhere in between the devices we have today and perhaps an early fault-tolerant machine. So now let me talk about a slightly different idea, which might be more achievable in the near term. And that's averaging with the continuous ancilla degrees of freedom. And so the context I want to focus on here, even though the idea can principally be applied to solid state NMR as well, is I want to talk a little bit about EPR experiments, electron paramagnetic resonance. And this is also a technique used in various systems to study catalysts, photosystems, molecular conductors, as well as proteins in a live cell environment. And that's kind of the context I want to keep in mind for the next few slides. And this is a case where, let's say I have a protein and I want to see how it behaves, but of course I care about it as to how it behaves in the environment that is relevant, right? Which is in a cell, in a live environment. Perhaps I want to understand its biochemistry there. Perhaps I want to see how different, drug candidates or behaviors operate. And solution NMR in many cases may tell me the relevant information about the system, but there are several contexts in which studying the protein's behavior in a solution or even in a solid state experiment separately predicts something very different than what happens in an actual cell. And this is a case where then you really want to see how the protein behaves in its true environment. This becomes quite difficult. You can't use things like X-ray crystallography or even cryo-electron microscopy in these cases. Standard NMR also becomes a problem in this case. And so what people are starting to do, this is a relatively new technique as far as I understand, in terms of how widespread it's being used, is to apply spin labels onto your protein. So I synthesize these proteins with radicals that I have labeled different parts of the protein with, and these are active electron spins. So these are these blue kind of bubbles on my protein over here. And then I will let that protein kind of go about its life inside of a cell. And while it's doing this, I will do a magnetic resonance experiment on these spins that I can now access with my different control coils. And the one protocol I put down here is something called DEAR, double electron resonance, or a simplified version of that. And you can think about this as I treat one spin as a probe and the other as a pump, or observable in a pump here. And I do a spin echo on one of the spins, which basically cancels out, hopefully, the effects from any other spins or magnetic fields or things in its vicinity. And if in principle would cancel out the interaction, the dipolar interaction it has with the second spin as well. But which acts as an effective magnetic field on my observable spin. But then what I do is at varying times, I apply an inversion pulse or a pi pulse on my pump spin. So I act as if I am changing basically the relative duration of time that second spin acts like an effective magnetic field on my first one. And so the echo from that second spin is not fully canceled out. And of course, like the signal I will get then in this will be modulated by the frequency of the interaction, the strength of the interaction between the two spins. And that strength depends on the distance between them. And that's really what I'm trying to infer. So you'll get an echo that looks like this, echo intensity that looks like this with some sort of dephasing. And the modulation of this echo will tell you information about the relative distance between the nuclei. But the thing really here is like, I really need to be able to simulate the spin dynamics to extract this distance distribution. And that's the thing, it's not a single distance, sadly, in many of these experiments. What they'll do is they will very rapidly cool down the live cell and things will freeze in slightly different orientations and the various copies of the protein in the cell. And so I'll actually get a distribution of the distances. And it's from this distribution and appreciable changes in it that I will see what happens in the cell, like as this protein is used, right? So you can imagine a case where I send in the protein and let's say nothing happens, then I'll get a particular distribution. But let's say it is acted on by and manipulated by various mechanisms or processes in the cell, the distribution may change dramatically. And this is what people use to infer information about. But I need to do simulation of this magnetic resonance experiment to actually extract this distribution. The second thing is that while this simulation is almost analytically solvable for spin halves, which are very commonly used spin labels, this can be hard to do for spin labels that has spin greater than 1/2. And the difficulty is not because doing a simulation of 2 spin 7/2 by itself is any big challenge. You can do it on your laptop, but there's a massive amount of ensemble averaging that needs to happen. And you can ask, well, why do I care about doing higher spin simulations, higher spin experiments? And I'll mention that a little bit later, in which it actually turns out for chemical reasons, biological or chemical reasons, higher spin labels tend to survive inside a cell for longer times and be more inert and avoid getting garbage collected by the cell. And so you can actually do longer experiments and learn more about the system you care about. So let's try to understand this a little bit more concretely. This high spin EPR experiments. So again, I have let's say a protein with 2 spins between them, and let's assume a particular context, which is common, of high fields. So high extra Zeeman field that's applied, magnetic field that's applied to the system, an isotropic G tensor, which just means that the spins behave, their local magnetic response, this SZ term, is isotropic, and then a pair of spins that have spin higher than 1/2. So now I have a Hamiltonian that looks like this, and the various terms are this local magnetic response, this SZ term. Then this SDS term is the zero field splitting that's there in the system. And generally this D is a 3x3 tensor. Then I also have this SAI term, which is the hyperfine interaction between the spin and the nucleus. And then the direct dipolar interaction between the 2 spins, which is what I'm really trying to learn from. And the issue here is that many of these parameters in the experiment actually have an ensemble distribution. So first is the orientations themselves. This is our powder average that we talked about in the solid state NMR context. And so there's at least one parameter here which you have to average over. The second is that the eigenvalues, for example, of this zero field splitting are also distributed, and you generally don't need to look at all 3 eigenvalues. You can kind of codify them into 2 parameters. But now you have 2 spins, so you have 2 parameters each and 4 total. And then, of course, the distances themselves, which are what you're trying to learn, are distributed. So at the very minimal amount, you have 6 independent parameter distributions. And so if you look at the typical grid sizes that people use for this, you might need millions or tens of millions of points to repeat this simulation. And so even though each one is trivial, relatively trivial, and of course, if you had access to very powerful supercomputers, this is definitely doable, like with modern day, computers. For the, average, experimentalist, like doing this, they may not have access to these resources or know how to use them efficiently. And it becomes challenging. And really, like this, even with the access to some of this equipment, people have avoided doing higher spin label EPR experiments for things that are larger than spin half, like largely because of how onerous this simulation is. And I'll give a few more details about why you might want to use these higher spin labels. And the answer is really that these cells are very good at, for example, garbage collecting radicals and throwing them out of your system, for example, to prevent cancer and things like this. This is, I guess, the evolutionary mechanism for it. So if you use different metallic compounds instead as labels, especially coated, I guess, in some particular ligands and stuff to prevent toxicity, these tend to last a lot longer in the cells and are more biologically inert in that sense. And so these are cases where people have used a few of these higher spin labels, these metallic compounds so far, that for particular reasons behave as effective spin halves. So they try to get away with using the same simulation and analysis they do on spin halves on that. Even there, sometimes you have to be careful and really need to do a higher spin simulation. But there are other compounds, other labels that might be more biologically desirable, that last longer in your cell, that you really need to treat as higher spin and to do simulations on, but people don't do these experiments, even though they might be nicer, because the simulation is too annoying. And so this is kind of the context where we're going to try to target. And I will just very briefly sketch out an idea on how to do this with a bosonic ancilla, and then details are I have more than I've shown here, but they're not finalized. So I'm happy to talk more about it in words if you have questions than formal slides. But the basic idea is that the key ingredient is, let's say I have a system of spins, in my quantum simulator or device, as well as at least one bosonic degree of freedom. The common contexts to keep in mind for this could be, let's say, a system of trapped ions where the phonon, that's there in the chain, that's normally used to mediate interactions in any trapped ion computer or simulator, can be used as a bosonic mode directly for simulation instead. Another common context that is becoming, I think, very hot recently is use superconducting platforms where you can take microwave cavities, resonators, and couple them to transmon qubits. I think the folks at Yale are doing a lot of interesting work on this in the context of error correcting codes, continuous error correcting codes. I think Steve Gervin came and gave a talk last week at here. So the key point is, if you can synthesize an interaction where the boson quadrature, which I've listed as X here, controls a spin interaction, one of the ones I showed in the previous slide, then you prepare the system in kind of a trivial product state where the spin is put in the standard computational basis state that you might want to, as per the prior simulations. But you can prepare the bosonic state with a wave function that is essentially whose amplitudes are the square root of the distribution you're trying to average over. Now, this can be a non-trivial thing. It is a detail that has to be worked out, but there are cases where you can imagine that perhaps, depending on the distribution you're trying to average over, they tend to be fairly well behaved in some of these EPR contexts. And so maybe you can try using things like Gaussian mixture models or whatever it is to fit the distribution. And then each Gaussian in that mixture model can be prepared as a display squeeze state of the boson, which is something that we know how to do in principle, and there have been experimental demonstrations of. So then, the way you get your ensemble averaged observable is, what you'll measure is, again, this type of thing, the expectation value of total magnetization over as a function of time. And because you start in a product state and you don't change the wave function directly of the boson or operate on it, the bosonic operator will turn into a classical number x that is weighted by that initial distribution wave function of the bosons. And so now what you get here is this, this X, which acts as your classical variable that you want to average over, and you get your ensemble averaged free induction decay. So as I mentioned, one context we're thinking about doing a proof of principle experiment of this for still perhaps in spin halves or maybe spin one or something like this, are systems of trapped ions where they naturally can realize things like this controlled displacement using these phonon-mediated interactions, and then combining that with a couple 2-qubit gates that are standard can turn this into, let's say, a boson like this X-controlled Ising-ZZ interaction, which actually turns out to be one of the main ingredients for many of these EPR simulations, as the dipolar interaction in high fields really kind of turns into that in many common contexts. You can think about doing it for higher spin as well. I think a similar type of interaction shows up naturally, as listed in this PRX reference, I think from Chris Monroe's group. I put in the bottom. But superconducting, platforms are also a place where people are developing great toolboxes for this type of work. You can even imagine a case of Rydberg atoms where they don't have a natural bosonic mode that shows up, but I think very recently people have been playing with putting Rydbergs in microwave cavities to look at extend lifetimes and things like that of different Rydberg transitions. But perhaps there similarly, if you write down the light matter Hamiltonian, you can maybe use the microwave cavity itself as a simulation variable, right? Here, when you use the same tricks you use to prepared state that people are used to in superconducting cavities. So this is the basic idea, and at least with initial simulations, it seems to work. But of course, working out the details and doing a proof of principle is something we're still doing. So just kind of in conclusion, to illustrate the main points that I was discussing today, we have NMR, which really is kind of a Hamiltonian inference or dynamical inference problem is a way to frame it. And being able to simulate the spin dynamics given a particular Hamiltonian can turn out to be a very useful thing that in some contexts is classically hard to do from a practical perspective. And the hope of using quantum devices to do this is one that you can do faithful time evolution in quantum hardware, even one that's not fully error corrected or fault tolerant in principle, as long as the decoherence in the quantum hardware is less than the decoherence in the NMR experiment. This still may not be such a simple thing to do, but at least you have a finite tolerance for error in your system, which is not true for many other Hamiltonian simulation contexts that are people are thinking about. And the second is that you might be able to use ancillas in a superposition to do efficient ensemble averaging in these cases, and there are some of these ideas that we're working out for that. So with that, I will just acknowledge all the various collaborators, both on theory and experiment, as well as Ilya Kuprov and Hariyar Thanari, who are folks solidly in the NMR and NMR simulation communities who've been extremely helpful in teaching physicists what about the biological context and prior literature in this field. So with that, thank you, and I'm open for any questions.
Well, thank you very much, Kushal, for a beautiful overview of NMR simulations. I think that it maybe is good to stop your screen sharing so we can all see each other and open the floor to questions. So I'm sure there's going to be a great deal of discussion, and let's start it now. So please turn on your video when you talk so we can have a back and forth.
Hello.
Marius, you'd like to start?
Yes. Can you just go back to his last slide with this boson, extra boson term?
This one?
Right. So, or maybe, yeah, exactly this slide. So I just want to mention that on a theoretical point, this kind of averaging over this e to the ith spin, that is actually simulating an open system, which has been studied in terms of a Lindbladian, in particular if the p of x is a Gaussian distribution. So you get a new generator of an open system, and these guys have very strong decay properties. They are better than the Hamiltonians. So if you want to have any talk about this, we are studying this forever, and we have always, people looked at this, why do you do this average from a theoretical point, and you're not talking about Hamiltonians anymore, and you get this Lindbladians? We just love the Lindbladians for some other reasons, right? So if you want to talk about it, there's a lot of theory. If you use this averaging, particular for the Gaussian distribution P of X, you get a ton of decay properties which are faster. And knowing this in advance could probably even help with some speed of simulation, right? If you know that decay to equilibrium is fast, and these guys are easier to handle than the single Hamiltonians.
Okay, that's very good to know. No,
if you want to send me an email or whatever, or we chat, it's just that I recognize the term. So this is, why these seminars are great. So I recognize the term. I'm very happy. And I can now call this, additional Bose continuous variable ancilla thingy, right?
No, I would love to chat. No, that's great. It's also, as you said, useful, these seminars, to connect with other people who have been thinking about, the same problem from different angles.
So a successful seminar leads to a new collaboration. That's great. That's really great.
Good. Thank you.
Yep.
Anybody else?
I'll be around during the day at Harvard if people want to chat later as well.
That's great. So are there no other comments? Oh, it was really beautiful talk. You outlined many of the ideas, and we'll look forward to seeing some of the details when you post your new papers. So thank you so much, Kushal. It was a great talk.
Thank you, Arthur.